Over the past few years, several new biocontrol agents have been commercialized in Canada. In this video, we will introduce you to two of them. We will talk about their place in biocontrol programs, their biology, predation habits, and the best ways to use them. The information is based on the research done at Vineland. Although we already know a lot about these new biocontrol agents, there still is a lot to figure out. In the past, new biocontrol agents were developed in a very targeted manner, introducing one or more specialized predators or parasitoids for each pest species. However, in recent studies, the value of relatively large generalist biocontrol agents, also known as big bios, that can attack a wide range of pests is increasingly recognized. And these are the big bios found and studied here in Vineland and commercialized by the company Applied Bionomics. First, the red one is Anistis baccharum. Its common name is the whirligig mite, but we have nicknamed it crazy mite. The second one is Navis americoferus. Its common name is the dunzo bug, but again, we have nicknamed it ninja bug. For decades already, both species have been known to be good biocontrol agents in outdoor crops. However, the lack of effective mass rearing methods, due to their voraciousness and cannibalism, prevented their use as biocontrol agents in augmentative biocontrol programs until now. Our research solved this problem, and both species are now available to growers. This figure shows our version of a food web in a greenhouse crop. Our new big bios are in red circles, and it shows why they are important. The blue lines shows the previously known relationships, and the red lines are the relationships our researchers uncovered. From bottom to top, we see the crop with multiple pests that are managed with several small specialized predators and parasitoids. The big bios at the top can strengthen biocontrol programs because they will control multiple pest species at the same time and multiple life stages, including adult stages of the pests. Also, their diverse diet will allow them to persist in the crop. This means that they can be released and established in crop before pest infestations occur, making the system resilient to pest invasions. It also means that they won't have to be released on a weekly basis, which may cut costs and reduces the dependence of the biocontrol program on the whims of the carrier delivery companies. Although the big bios can eat other smaller biocontrol agents, multiple studies shows that the biocontrol efficacy tends to be greater in systems with big bios. There will be a bit more about this later in the video. Because of their voraciousness, it is relatively more expensive to produce them. However, good news is that they can be produced on-site by growers using aphid bunker plants. Overall, the big bios have a wide range of pests they can attack and often take on new invasive pests without hesitation. First, let us introduce you to the Anistis mites. These are very aggressive predators. They rapidly search all the foliage surface until they encounter something to eat, such as aphids, thrips, and other mites. We have seen it prey on pests that are not well controlled by current biocontrol, such as echinothrips, mealybugs, psyllids and plant hoppers, and whiteflies. They can prey on almost all developmental stage of pests, including adults, complementing other biocontrol agents that cannot. They will kill and consume pests, but they also harass, preventing pests from feeding, egg laying, etc., contributing to overall control. They will also attack and kill more prey than they can eat. Finally, they feed on pollen and several supplemental food sources such as Ephestia eggs and Artemia cysts, like in this picture. Knowing the life cycle of anistis is important to determine if the released mites are reproducing and establishing in your crop. The individuals in this species are all females, which can lay eggs without mating. Eggs are typically laid in loose-growing media or concealed spaces. After hatching from eggs, the first motile stage is the larva, which can be distinguished from older nymphal stages by the number of legs. The larvae have only 6 legs. Their size is about quarter millimeter. The next 3 motile stages are the nymphal stages. The only difference among them is their size. Protonymphs are about half a millimeter, deuteronymphs are about 2/3 of a millimeter, and tritonymphs are about 1 millimeter and a half. Each motile stage is separated by a pupal stage called a chrysalis. Probably the most important stage is the adult, because their greater appetite and longer duration. The adults are about 2 to 3 millimeters and live about 3 weeks. Once they become adults, they are ready to lay eggs within a week, and each female can lay up to 5 egg masses, and each egg mass contains about 25 eggs. One concern with generalist predators like anistis is that they would also eat other biocontrol agents. To investigate this, we assessed the compatibility of anistis in lab trials with Bavaria bassiana, an insect pathogenic fungus, and with smaller predatory mite species on foliage, and with a soil-dwelling predatory mite, Stratolepis semitis, and with a whitefly specialist lady beetle, Delphastus catalinae, and with another big biogeneralist, Aureus insidiosus. In the petri dish, adult anistis mites attacked and fed on smaller phytoceate predatory mites, but their mortality was reduced in the presence of alternative food source. Adult Aureus, Stratolepis, and Delphastus were rarely attacked. Finally, anistis was compatible with Bavaria residues. Several greenhouse trials were conducted to test the performance of anistis as a predator against common greenhouse pests. The graph on the bottom left shows a greenhouse cage trial with western flower thrips. In this trial, there were 4 chrysanthemum plants in the cage, infested with thrips, and then put either 2 cucumerous breeding sachets or 8 anistis mites or a combination of the 2 treatments. Over the course of 8 weeks, you can see that the treatments with both mite species by themselves performed very similar, the blue and red lines. But the best thrips control was obtained when the mite species were combined, the green line. Another greenhouse trial investigated the mite's potential to suppress foxglove aphid population, and the result is shown in the graph on the bottom right. The benches in the greenhouse were filled with pepper plants, which were infested with aphids. Then the aphid parasitoid, Aphidius arvi, was released in the whole greenhouse at the rate of 2 parasitoids per square meter. Half the plants also got 1 anistis mite per plant. The result shows that the best and fastest control was achieved on the plants with the combined parasitoid and anistis treatment, the red line, as compared to the aphid parasitoid alone treatment, the broken line. The peppers also suffered an accidental infestation of thrips, which anistis controlled very well, and as a result, pepper yield on the plants with anistis was increased by 15%. One interesting application of anistis may be in cannabis production. We have observed it eating root aphids, but have no idea yet if it will control them. As a first step, we did a trial with Niagara College to see if anistis would even like this crop. Anistis mites did establish, reproduce, and complete their development on both crops. Similar numbers of mites were observed on both cannabis and chrysanthemum during weekly observations. Other crops that anistis may do well include greenhouse berries and some outdoor crops. As a new product, the best rates and release methods are still guesswork. Overall, we have observed good results with just a few anistis per square meter. The first option is to establish anistis in the preventative way. For this, you would release one or a few adults per plant and repeat this 2 to 3 times, depending on the pest pressure. After a single release, a new generation will start emerging 4 to 6 weeks later. The use of supplemental food such as Ephestia eggs, Artemia cysts, or even cucumerous mites will help the predator establish the population. The wide host range of anistis will allow it to persist in the crop. The second option is fast suppression of hot spots with releasing anistis at a higher density. In general, anistis will tolerate both low temperature and low light, and it prefers the humidity above 50%. Although anistis is found in many types of plants in the wild, including herbaceous, grass, shrubs, and trees, our preliminary observations indicate that anistis may not establish in tomatoes due to the granular trichomes in the stems, or in plants with smooth and slippery stems like roses and poinsettias. The feedbacks from commercial growers are showing the potential of anistis mites. Because of their erratic, aggressive behavior, the anistis might also disrupt pest populations through harassment. When anistis runs through a patch of pests, it scares away the pest, causing them to fly away and get stuck on the sticky cards. It is recommended to run a sticky tape floating above the foliage to maximize the capture of flying pests but minimize the capture of anistis. In a commercial pepper greenhouse, anistis was released preventatively and they persisted until the end of crop cycle. Anistis keeps feeding and reproducing at cold temperatures, making them a great choice for biocontrol during the winter months. The growers are impressed with immediate predation, but the mites often fail to establish on the crop. So when using it preventatively, it is recommended to have plenty of alternative food available. One cheap way to accomplish that is to release cucumerous mites together with anistis mites. The cucumerous mites themselves are effective biocontrol agents and also serve as food for anistis mites. For the moment, anistis is only available within Canada. The species Anistis baccharum is described as a cosmopolitan species, which means it is found all over the world and has the potential to be commercialized globally. We are working with taxonomists in Canada, US, and Europe to confirm this. Currently, it is not sure if all Anistis baccharum found in different areas are truly the same species. Unfortunately, this process takes time, so for now, the Canadian growers are the lucky ones that get to play with this exciting new biocontrol agent. Now, let us introduce you to the next predator, Naves bug. Naves bugs are abundant in gardens, orchards, and field crops. They are voracious predators of pests like aphids, spider mites, psyllids, thrips, and butterflies and moths seen in the top picture, and tarnished plant bugs seen in the bottom picture. Both the immature nymphs and adults are predaceous, and they search actively for the prey, which consists mainly of other insects and mites. Navis bugs are swift predators. They catch and hold prey with their forelegs like similar to mantids, then rapidly suck the body contents from their prey. They also feed on Ephestia eggs and Artemia cyst, which can be used as supplemental food source for preventative establishment of the predator in the crops. Here is the life cycle of Navis, which will help to determine if the released predators are reproducing and establishing in your crop. The young adult males and females usually mate within a week after their final molt. Females insert their eggs individually into plant tissues. After the eggs hatch, the nymphs go through 5 immature stages. All nymphs resemble the adults, only they are smaller and lack developed wings. The young nymphs have tendency to stay close to the soil surface, hiding under dense foliage and debris, which is likely to avoid cannibalism from older and larger individuals. Because of their smaller size, the young nymphs also prefer smaller prey such as thrips and mites. The nymphs molt from one stage to the next quickly, which is usually completed within a few hours. Navis bugs develop from eggs to adult in 4 weeks. The adult stage is the most important as they are the most voracious and live for 4 to 6 weeks. Females lay 4 to 5 eggs per day for a total of 100 to 150 eggs over their whole life. The efficacy of the Navis bug was compared to its close relative, Dicyphus, another big bio that's been commercially available for more than a decade. The pictures show their relative size. Only adult females were used for this study. The Navis was better than Dicyphus at killing larger, more mobile prey, such as adult aphids, adult whiteflies, and adult thrips. Both predators did well against thrips larvae, and the Navis was less efficient at killing smaller or sessile prey such as spider mites and whitefly nymphs. The compatibility with other biocontrol agents was assessed for the Navis bug as well. The tests were done with Bavaria bassiana, an insect pathogenic fungus, and with smaller predatory mite species on foliage, and with parasitic wasps for aphids and whiteflies, and with 2 other big bio generalists, Aureus and Dicyphus. In the petri dish, adult female Navis were compatible with Swarovski, Persimilis, and Encarsia, but did prey somewhat on Aphidius collimani mummies. The Navis significantly preyed on the other big bios, both Aureus and Dicyphus. The insect pathogenic fungus Bavaria bassiana caused high mortality in the Navis bugs. Several greenhouse trials were done to test the performance of Navis bug as a predator against common greenhouse pests. First, we did a few trials on peppers and tomato plants. The Navis alone did reduce the pest, but did not control them completely. It is therefore best used in the combination with other biocontrol agents. The graph at the bottom shows that the best establishment and pest control were observed when the Navis adults were released preventatively with aphid bunker plants. These trials used only one tall plant with large leaves in a cage, and we noticed that the Navis did not seem to be attracted to the plant. Navis is a shy predator, preferring to hide in dense foliage. Considering their plant preference and their natural habitat, we decided to try another crop type next. Therefore, the next crop the Navis was tested on was greenhouse strawberry. The goal was to determine the value of adding a Navis bunker plant system to a standard biocontrol program based on predatory mites to manage thrips and spider mites, evaluating pest suppression and compatibility with predatory mites for 12 weeks. In the standard treatment, Californicas and cucumbers sachets were placed on potted greenhouse strawberry plants. The mite sachets were replaced after 6 weeks. The standard plus Navis treatment used the standard mite sachets and 2 pairs of Navis adults on the bunker plant. The last treatment, which was the half standard plus Navis, was similar to the second treatment, but the mite sachets were not replaced. The pests were introduced in 2 waves, at week 0 and halfway at week 6, as seen in the timeline figure. The results were fantastic. Navis bugs were definitely more affiliated with strawberry plants compared to peppers and tomatoes in previous trials. They initially reproduced in the bunker plants with aphids, but quickly spread among the strawberry plants. For thrips and spider mite control, the standard predatory mite program plus Navis did the best, the red line. But what's more notable was the half-standard program plus Navis, which is the green line, did better than the standard predatory mite only program, which is the blue line. And neither the predatory mite population nor the Navis population was affected by the presence of one another. Therefore, it was concluded that the addition of Navis americoferus to greenhouse strawberry IPM program based on the predatory mite sachets is beneficial. As a new product, the best rates and release methods are again still guesswork. The first option is to establish Navis bugs in a preventative way. For this, you must have aphid bunker plants. To ensure the adults mating, it is a good idea to release a few adults on each bunker plant. You can adjust the number of these Navis bunker plants per area, depending on the pest pressure. After a single release, a new generation will start emerging 2 to 3 weeks later. The use of supplemental food such as Ephestia eggs, Artemia cysts, or even cucumbers mites will help the predators to spread onto the crop. Their wide range of potential prey items will allow the Navis to persist in the crop. The second option is fast suppression of hot spots by releasing Navis at a higher density. The Navis will tolerate low temperatures. In nature, the overwintering adults can be seen walking on the snow on a sunny winter day. Navis americoferus prefers crops that are closer to the ground, such as alfalfa, soybeans, and grasses. And the females will not lay eggs if they were exposed to short day lengths while they were nymphs. And from our experience until now, here are the greenhouse environments that will be unsuitable for either Anistis or Navis. The big bios are voracious and always on the search for food. A small, isolated plant seldom has enough food to keep the big bios sustained. Therefore, small, isolated plants on flood or mesh top benches are not compatible because the predators will wander off the small plant and get drowned in a pool of water or get lost as they go down the bench and reach the ground. The mesh top bench with drip irrigations will be fine as long as the crops are mature and in contact with one another. the predators use the foliage and the irrigation lines as a bridge to walk across the crops. Both predators prefer dense foliage and do not like disturbance of their habitat, so they are not suited for plant stages that are small or young and to be transplanted or moved around. They are also very sensitive to direct contact with soap and oil product. So what kind of environment then is friendly to the new big bios? They will like it where crops are directly on the ground with pot-type placement, especially with young plants, or mature plants in contact with one another. Hanging troughs work well too because it often provides a foliage-dense environment. As mentioned earlier, they do not like their habitat disturbed, so ideally they should be released in long-term crops that do not get moved around, such as vegetables, fruits, and cut flowers. And thank you for watching. Hopefully, this information will be helpful to you when considering to incorporate the new big bios into your IPM program.